Hello students, I am Dr. Meenakshi Mahur, Assistant Professor, Zoology, Sri Samlaji Government College, Mandvya, Chittorgarh. Today my topic is Canal System in Porifera. So first, what is canal system? It is water circulatory system of sponges. Canal system is the distinguishing feature of all sponges. In sponges, there are perforation of body by numerous apertures, which is used for the ingress and egress of water currents. And in them, the water current flow through a certain system of spaces, which are collectively form canal system. The most vital role in the physiology of sponges is played by water current system. All the exchanges between the sponge body and the external medium, that is water, are maintained by means of this water current or canal system. Food and oxygen, which enters into the body, and the excreta and reproductive bodies, which are carried out of the body, is through the canal system. And this is also used for causing beating of flagella of collar cells in porifers. So this is canal system. In porifers, there are three types of canal system. The arrangement and the complexity of this system vary considerably in different sponges. They are of three types. First is ascon type, second cyclone type, and the third is leucon type. So first of all, I'll explain ascon system. It is the simplest type of canal system and it is found in asconite sponges like Glucosalinia. So here I will explain this canal system in Glucosalinia. In it, the body surface is perforated by large number of minute openings called incurrent pores or ostia. So here you can see these are the dermal ostia. Here these are dermal ostia or incurrent pores. These pores are intracellular spaces within tube-like cells called porocytes, and they extend radially into the mesenchyme and open into the spongocyte. So see, these are dermal ostia and they open into spongocyte. This is a spongocyte. The spongocyte is the single large spacious central cavity in the body of sponges, and this is lined by flagellated collar cells or coinocytes. So these are coinocytes. So spongocyte is lined by coinocytes, the yellow one. No? These are the flagellated cells. Spongocyte opens to outside through a narrow circular opening which is called osculum, which is located at the distal free end, and it's often fringed with large monoaxon spicules. So how it works? The surrounding water, if you assume that this is water, it enters the canal system through ostia, from ostia. So it enters through ostia, and the flow of water is maintained by beating of flagella of collar cells. This rate of water flow is slow because the large spongocyte contains much water which cannot be pushed out readily through single osculum. So first water enters through dermal ostia, then it comes into spongocyte, and then through osculum water exits out. So this is the pathway: ingressing water, ostia, then spongocyte, osculum and outside. So if this is a dermal ostia, water enters into the body through it and comes into spongocyl, and then from spongocyl it comes out through the osculum. So it is a very simple canal system, and it is known as Ascon type canal system, and it is found in the asconoids like Glucosalinia. Now next, Sycon type. This system, this Sycon type of canal system is a more complex system of pores and canals, and it is found in syconoid sponges like Sypha, which is also known as Sycon, and Grantia. So here I am going to explain canal system, Sycon canal system in Sycon or Sypha. So uh, in this body wall of syconoid sponges includes two types of canals, that is incurrent and the radial. So here you can see this is a incurrent canal here, and then there is a radial canal. This is an incurrent canal and this is a radial canal. They are paralleling and alternating with each other. So here you can see incurrent, then radial, incurrent and the radial, incurrent, radial. So they are present parallel and alternate to each other. And both types of canals end blindly in the body wall, but they are interconnected by the minute pores, that is incurrent pores or dermal ostia, which are found on the outer surface of the body, and they open into the incurrent canal. So these are the dermal ostia. These incurrent canals are non-flagellated, and they are lined by pinacocytes, and they lead into adjacent radial canals through minute openings called prosopiles. So here are prosopiles. Uh, it is not clear that prosopiles are channeled through porocyte, but it is a certain that in the adult they are simple intracellular spaces. Radial canals are flagellated chamber, as only they are lined by coinocytes. They are lined by coinocytes. These canals open into the central spongocyl by internal ostia or apopiles. So here are apopiles. These are apopiles. These are apopiles. Spongocyl is a narrow, non-flagellated and cavity which is lined by pinacocytes. It opens to exterior through a excurrent pore, that is the osculum. It is same as that of Ascon type. So course of water current can be represented like water enters through dermal ostia and comes into the these are the dermal ostia. So water comes into the dermal ostia, then moves into the incurrent canal. Then incurrent canal through prosopile, these are the prosopile, moves into the radial canal, and then from radial canal they moves into the spongocyl through apopyle, these are apopyle, and then they moves outside through osculum. Again I'll explain. First, ingressing water, these are the dermal ostia. So it moves into the incurrent canal, then in incurrent canal through prosopile, these are the prosopile, moves into the radial canal, then these are the opopiles, and so through opopiles water moves from radial canal to spongocyl, and then it goes out through osculum. So this is a Sycon type of canal system, and it is found in cycle. Then third is Leucon type. Uh, because of the more folding of body wall, the cyclone type give rise to a more complex canal system, the leucon type. And this is found in leuconoid sponges, like spongosil. So I'm going to explain leucon type of uh, uh, type of canal system in spongosil. So first, radial symmetry is lost and the canal system has become more complex. Here I'll explain. Here you can see there are dermal ostia. These are dermal ostia. So ingressing water, that is water from outside, moves into the body through dermal ostia and it moves into the incurrent canal. These are the incurrent canal. Then it moves into the flagellated chamber. These are all are the flagellated chamber. But from incurrent canal, water moves into the flagellated chamber through prosopiles. So these are prosopiles. These all are prosopiles. Then from flagellated chamber, water moves into the excurrent canal. These are the excurrent canal. But water moves from flagellated chamber to the excurrent canal to the apopiles. So these are the apopiles. From excurrent canal, water moves into the osculum and it comes outside. So this is a canal system in leuconide sponges, and it is known as leucon type of canal system. It appears to be the modification of cyclone type, and it is found in many calcareous sponges. Leucon system is developed directly without passing through the ascon and the cyclone type in their embryology. In demospongy, leuconite condition is derived from a larval stage called the rhagon. And the spongocele of rhagon is surrounded by flagellated chamber opening into it through very wide apopiles. In them, a single osculum opens at the top of the spongocele. So these all are about canal system. So first I described ascon type, then cyclone type, and the third one is leucon type. Ascon type is the simplest one and the leucon type is the most complex one. So these all are about canal system in Porifera. Thank you.